Testing. There we go.
Thank you.
So once again, we'll reprise that question. Who is ready to go to Mars?
Woo!
So you think you're ready to go to Mars. Are you ready to stay on Earth? 1 and 2. All right. All right. That's a good series. I want this to be interactive here. I am not a medical doctor. That's my disclaimer. I find myself frequently on panels with doctors, so I am not a medical doctor. I will not make any diagnosis or prescriptions. If you see something here of value to you, see your family doctor, which I think we have a few in the audience, which is a good thing.
So you've introduced yourself?
I have introduced myself.
So we will.
We're good. We're good.
Before you continue,
Sure.
I wanted to mention that on all of your schedules, there's an open slot at 3 o'clock, and we actually have a speaker at 3 o'clock in this room on the medical track. Doug Plata will be coming to speak about extending crew stays on Mars missions and moon missions. So it'd be great if you're interested to attend.
You have to.
Sorry for the interruption.
That's all right. Okay. Here we go. No, don't touch anything else. Just hold on this side. Yeah. Okay. Anyway, okay. So, but I've been a professional chemist since 1981. We have a family business where we do water, wastewater testing. It's an environmental analysis firm, so we deal a lot with the issues that expose you all to things that compromise your health. And so that's why I can say with some degree of confidence with my experience and both professionally and as a father of 4, that there's a lot that we can get done on Earth that you can do for yourself to get ready for this trip to Mars. And by the way, it might help you to stay on Earth as well. A personal note, the program is dedicated to my son, Kevin William Gardiner, who died in September 30th, 2014 by his own hand after many years of psychiatric care and health issues caused by that. I emphasize the Tuesday. It's the Teutonic god of war, Teus, and that's the rune for Teus on the right, and that is a little mythological depiction of Teus, eaten by Fanfur, and his right arm is being eaten there, which is maybe a reference to something in the program, which I'll talk about a little bit later. So, Georgia, where I come from, was a noble experiment. It was a penal colony to try new social advances in the New World. So we can do that on Mars as well. We can establish habitats on a planetary surface where the essential conditions for sustainable life will be enabled by the proper design. And earlier speakers, like Mr. Maltz here, did that. Will be enabled by proper design maintenance by the founders and settlers. And luckily we have a great guy here who's doing this. Now, I don't know if anybody's given much thought to this concept, but the large separation in space between Earth and Mars will both necessitate and enable provision of the conditions for indefinitely sustainable human life on Mars. Well, that's kind of an idea that we all have. But here's something else that is food for thought. The frontier nature of the initial settlements removes the disincentives for reforms to a current pharmaceutical and medical practice and provides a platform for innovation. And by that I mean, I'm going to show you health concepts here which will help define a baseline health activity that if it's successful will largely keep you in a healthy condition that would not so often necessitate medical interventions. So a controlled ecological life support system the previous speakers did speak about will include air, water, nutrients, waste handling, energy input management, and notobiotic design. Notobiotic is a word biologists use to describe a complete description of species in a given habitat or microorganism. And it'll have a custom design consistent with the health status of the settlers. So we'll have to do a lot of baseline studies on the settlers or the prospective settlers to determine their eligibility for treatment. So we've asked that question. 3-year round trip is the model, but consider that the known risks have changed. And I'm getting to the genetic part. So the known physiological problems for traveling in deep space for a length of time are, as previously mentioned bone demineralization and calcium loss, biological sensitivity to space ionizing radiation, visual impairment, and intracranial pressure, and astronaut ophthalmic syndrome. A lot's been said about radiation risk. Dr. Francis Cusinata, previously at NASA, now at University of Nevada, Las Vegas, is doing a lot of work on that. He's developed models to predict the degree of exposure to astronauts under various conditions and various models of space habitats. So actually, some of the things have already been pretty well addressed. Bone demineralization and calcium loss is considered by NASA to actually be solved. And they have done that largely with the advanced resistive exercise device. It's 2 hours of strenuous exercise aboard the spacecraft that has been shown in the astronauts who've done this and been monitored to actually work and reduce the calcium loss to a tolerable extent. There's been some other done work with diets, and I'll get to that a little bit later. We still have a problem with sensitivity to free radicals and oxidative damage. Well, the previous speaker had a very innovative solution to that in terms of the slide mold. But now it turns out that the ophthalmic changes in astronauts has been redefined. I'm just in receipt of a paper from Scott Smith and Sarah Zwart at Johnson Space Flight Center, who are the countermeasures branch of NASA and human factors, that shows that the cardiovascular, actually the VIIP, the intracranial pressure and visual impairment is not related to the fluid shift directly. It's not a direct result of the fluid shift, because the fluid shift does not cause an increase in pressure on the back of the optic nerve. They did parabolic flights with patients who had a port installed in their skull for the introduction of chemotherapy for brain cancer, and they were able to use the same port to perform intracranial pressures during parabolic flights, and they found there was no change in intracranial pressure. So that was ruled out as a cause for the visual impairment. But we are looking at epigenetics, genetics, microbiome, cardiovascular injury, and developmental plaques, weakening the vascular function and fluid balance. So geneticists talk about two kinds of genetics these days, in fact, in terms of the broad topic, and that is linear genetics or vertical genetics, the genes you receive from your parents, and horizontal genetics, which is a horizontal gene transfer between generally lower forms of life, but such as bacteria, which are very common sharers of genes. They transfer genes very frequently in our own gut. They're capable of sharing genes that supply survival value to other bacteria in other environments. And vascular status and fluid balance has become a major thing. Just recently, I encourage you to look up this website and keep an eye on it. I think you don't want to go to any better source than Harvard Medical School for a thorough discussion of space genetics. We've been urging this for years. I'm also chair of a committee in the National Space Society called Space Settlers Health Development Committee. And we've been urging the development of genetics for this purpose for some time, to protect human health, promote exploration, and sustain our own planet. I won't stay on this slide too long. It's pretty much been discussed about the epigenetics effects, the effect of food, and our gut bacteria. The favorable ones produce various nutrients that we need. There's a list of them here. So we already know that a healthy human anywhere has the natural genetic capacity to repair damage at the cellular level. And, but it's limited by carbohydrate metabolism and oxidative damage by acidity and ionized metals, which are available right here on Earth, and they have the same effects as ionizing radiation does in space. But a healthy immune system minimizes the load on the repair replacement system by preventing introduction of foreign materials and opportunistic microorganisms that causes cellular damage. but it's often compromised. There are an estimated 10 million mutations that occur daily within each of us. Yeah, but they're largely compensated for by these natural repair mechanisms. So we're largely unaware that these mutations are going on because our body takes care of it. We have, the human body, like all eukaryotic organisms, have a copy of our whole genome in each and every cell in our body, and that's trillions. So the question is, are we accessing them all, or to what extent do they back up each other? So this is the paper I mentioned I received from Johnson, from Scott Smith, Sarah Zwart. She's the lead author. So they're now calling this Astronaut Ophthalmic Syndrome. They've taken out the intracranial pressure part of it. And now they have measured many variables related to this issue. The International Space Station is a national laboratory, as you all may know. But it's also the most excellent metabolic laboratory you can imagine. It's ideal because of its isolation and because of the fact that they're able to quantify every possible input to the astronauts and everything they let out. And they can quantify the environment inside the space station, oxygen levels, contaminants in the atmosphere, carbon dioxide. All these things are directly quantifiable, which are very difficult to get here on Earth. You can do it, but it's all set up there in space already for the space station. So this is the new model that they've developed to explain astronaut ophthalmic syndrome, which is related to other things we have on Earth as medical conditions, glaucoma, for example. Intraocular pressure, I have a little bit of that myself. I've got to take drops to open up the channels in the trabeculi to maintain the flow of fluid into and outside of the eye. So this is what Sarah calls a multi-hit hypothesis. So you have not just one factor, but many factors causing this problem. The 1C genetics is the so-called one-carbon cycle. There's many metabolic systems in the body which utilize the one-carbon cycle, and it's a crucial cycle because it ties in with methylation, demethylation of genes, of gene expression. The previous speaker discussed. It's also tied into the production of neurotransmitters. It's tied into the the maintenance of cellular tissues, particularly in this model. It's related to endothelial tissues. So what happens though is that in the middle you have a schematized version of a blood vessel, okay, and so you have carbohydrate and insulin resistance, arterial stiffness, fluid shifts, radiation, and CO2. All are the various hits that we take on our systems here in space, and these have parallels on Earth as well, of course. So what happens is that they have found in the astronauts, particularly male astronauts, tend to have, the ones that have this problem tend to have a subactive allele that codes for methyltetrahydrofolate reductase, which is the enzyme that keeps the one-carbon cycle going, and the availability of the substrate materials for the maintenance and continued reconstruction of endothelial tissue. So with increased oxidative stress, that is with acid species commonly present in the diet, all these all other factors interfere with the efficiency of the one-carbon cycle, and in the presence of these subactive genes in the certain astronauts, particularly testosterone has effect on it too, they're just not utilizing their B vitamins to produce the endothelial tissue. See, so what happens then is that you have leakage from the veins in the subarachnoid space, which is a space between the pia mater and the arachnoid membrane that covers the brain. So, and this very narrow space extends into the optic cavity where the optic nerve extends to the eye, to the back of the eye. So, and there's also these what they call villi, the subarachnoid villi, which are projections inside this space. And with this fluid leakage, what's happening is that those villi are swelling. And so you get a kind of compartment syndrome here, where you have a buildup of local pressure that's not related to the intracranial pressure, but then that local pressure blocks the drainage of cerebrospinal fluid back into the back into the venal circulation system. So that's the proximate cause of these eye issues is a failure of the cerebrospinal fluid drainage, which is a downstream effect of the fluid leakage into the subarachnoid space and the swelling or edema, as the doctors say, of the subarachnoid villi. And this is a little bit more detail. I don't want to go into a lot of detail here, but in the properly functioning endothelial cell, what you're doing is using folate, which is basically vitamin B9, is running this cycle, okay? And that's the active form of folate, 5-methyl tetrahydrofolate. And that's driving the cycle to release nitrous oxide from endothelial nitrous oxide species within the endothelial tissue. So it's nitric oxide, NO. Not to be confused with nitrous oxide. I make that mistake all the time, so correct me. But that's nitric oxide causes the vascular relaxation, which is essential for the normal functioning of the cardiovascular system. But when that's blocked, when you don't have efficient production, the folate goes, you see it comes straight over here, bypasses this bioterin, this enzyme complex in here, which participates in the generation of the nitric oxide. It bypasses that, and you get reactive oxygen species like peroxide and these things over here, and these are highly reactive, so you get the oxidative stress and the fluid leakage. So, so here's some basic fundamentals of human physiology, which are very, very upstream and very, very pre-morbid, if you will, of medical conditions. And the first is that the blood pH must be maintained by the body at a slightly alkaline pH of 7.3 to 7.4. And the body closely maintains this pH level. Basically maintaining the oxidation reduction capacity of the body to perform all of its metabolic biochemical functions. And in the absence of that, respiration stops and you die. So, and it's the only, that pH is the only pH at which hemoglobin can exchange carbon dioxide with oxygen on the same molecule. It's quite a good metabolic trick for hemoglobin to do that. And so that has to be maintained in that level for it to work. And a little bit below that, you get metabolic acidosis, and it's a fatal condition. And in the case of most people and that's one of the early signs of kidney failure and liver failure and everything that emergency room doctors and intensive care doctors monitor very closely, because a little variation below that is a very bad sign and an early indication of a complete body system failure. So, but the good news is that you can get ahead of that with most food items are acid producing when digested. So this is an inherent stress in our diet, in the standard American diet, and I'll show you a chart later where all this started. So in the absence of these adequate dietary alkalinizing sources, the body resorbs calcium from the bones to neutralize the acids that have been picked up in the intestinal circulation around food consumption. Another interesting thing that occurs that blocks repair systems is that glucose, when it's present in circulation, causes insulin release and management of glucose at a fairly low level. I think the literature suggests that a level of as little as 25 grams a day is the level at which these effects occur. Insulin, the insulin response occurs. This particular work was done in a so-called model organism, another eukaryotic organism, there's C. elegans. You hear about C. elegans, it's just a little nematode, and it has a fast life cycle, so it's very good for testing. And what happens is that, so the glucose stimulates the release of insulin, induces glucose uptake, and the signaling inhibits this particular complex, this FOX0-DAF16 complex, which regulates the aquaporin 1 and other stress resistance and longevity dreams. So high glucose levels increase cellular reactive oxygen species, the peroxide and all that stuff I had in the previous slide. All that is increased in the presence of these, in the presence of insulin. So the 1972 USDA food guidelines helped us get into this mess, because it was based on a few tenuous studies done at Harvard, and it turns out that the data was cherry-picked and incomplete. So they didn't even check the carbohydrates. They saw a correlation between fat. This is Dr. Ansel Keys, who headed this research. He was the head of the Department of Public Health at Harvard at the time, as I recall. And he went to the McGovern committee that was working on this, the USDA, and they accepted it lock, stock, and barrel. So what happened was at the bottom of the pyramid is all these carbohydrate-rich foods, breads, cakes, rice, pasta, 6 to 11 servings a day. And going to the top where you had fats and oils to be used sparingly, and proteins and vegetables fell every place in between. So, but what happens now is that the overconsumption of carbohydrates and even the use of antibiotics promotes inflammation by promoting the growth of opportunistic parasites and organisms such as Candida. And Candida has turned out to be just a major system. There was a recent review of this, of Candida Research. This is very current research. It's very solid now. It's become a major field of research in medicine and health. So you get a failure of the tight junctions in the intestinal lining. So here's another really tricky human system. In fact, in every mammal that has any animal that has a digestive system, you've got to have a barrier here between your bowel movement and your blood circulation. Okay, and if that barrier is compromised, you get your bowel movement into your blood circulation. So I don't have to explain what that could do to you, but that's very common, and I'll show you a few slides of what that looks like when it's in your circulation. And that's commonly called leaky gut syndrome, or in the medical literature it's called luminal hyperporosity or intestinal hyperporosity. But upon healing, the tight junctions are restored, the inflammatory response is reduced, epigenetic effects are reduced, and autoimmune disease diminish. So this is just a schema showing that in process. So one of the things that I mentioned is the heavy metals. Bobby Kennedy Jr. had a press conference back in end of January showing that there were 250 studies showing that the danger or the harmful effects of ethyl mercury or thimerosal in the vaccines that are still has reduced quantities in the vaccines, but they still have to be used as a metal preservative in the multi-dose vaccine vials, because different people have different withdrawals from that same vial with different syringes, and the only way they can make that possible is to preserve it. And that's Eli Lilly developed the thimerosal in the 30s. They did a few rat lab tests. It didn't kill the rats right away. But we know now that it has a long-term effect, because what happens is that mercury takes the place of a complexing metal, selenium, in glutathione peroxidase. Selenium is an essential nutrient, and so when mercury is inside the glutathione peroxidase, it doesn't do its normal job of cleaning up the brain when you're asleep. It doesn't function, so your brain doesn't clean up. Heavy metals accumulate. One of the other metals that they commonly use in vaccines is what they call an adjuvant called aluminum. And where's that found? found in the brains of people who've died from Alzheimer's disease. So a nutritional biochemistry lab at Johnson, I mentioned Sarah Swart and Scott Smith. They started this study, which my company proposed back in 1986. There was already clinical literature on this. Not being a doctor, we got a principal investigator who was a pharmacologist who was a doctor, and we applied for this research grant, Small Business Innovation Research Grant from NASA. We weren't funded because Dr. Schneider, the head of the program, said he didn't think it was a valid technique because there was one VA hospital up at Hines did a study and they didn't show the effect. So he relied on that one result instead of all the other results from all the other clinical studies. I think you all know what pH amounts to in foods. Coffee, pH 5, wine 4, lemon juice 3, and then you go up on the other side, seawater, baking soda, and so forth. Yeah, oven cleaner, right. That's you wouldn't consume that, but it does. You want to stay away from it, in other words. So the body has, the metabolism of the body is very pH dependent. The hormones of the body function at the same range, 7.2, 7.4. So if you're constantly outside that range, you're outside the maximal functioning, optimal range of your enzyme functioning in the body. So, but with adequate oxygenation and good microvascularization, eliminating the compromise of the vascular system that I attribute to the discovery of Sarah and Scott at NASA, they have really identified this leaky vascular structures, veins, as a source of these issues. I'm a VIP red blood donor, and I have to take an iron supplement. And I wondered, why do I have to take an iron supplement? Well, I noticed one time when I had to really double up because I was didn't start the 2 weeks before like I usually do before a donation. My hemoglobin usually runs low. It's got to be 13, but I was running like in the 12s. And I took off my T-shirt after exercise one day, and it was red. It had actually, I apparently had actually leaked the iron out of my system through through my capillaries onto my T-shirt and through my skin onto my T-shirt. So, 1 minute, okay. So biomarkers of health and preclinical morbidity. There's pH is something you can check yourself. You can check your urine and saliva pH in the morning. The chart is marked with what you can you can measure it against to see you're in the right metabolic pH range. A live blood cell test is something that we're developing at NASA. I'm working with some Emory doctors and a student group to develop a research project to validate this, because people from the heartland of America, Tennessee, Georgia, have figured this out. And they're going out and taking, offering this service to people. It runs $35 to $50 a test. It takes 20 minutes. You do have to go in yourself, get a blood sample, a drop of blood sample, it's put on a slide, and you look at it, and you see what's going on there. A normal red blood cells, it's a little donut-shaped cell that circulates around. They don't stick to each other. They don't have any damage on the surfaces. But then you find that they do have some damage, like poikilocytosis. That's from heavy metals. This is my actual blood sample last time I had it taken. That on the right there is a uric acid crystal, and it's just like broken glass. That long tail is scratching the inside of my arteries and veins as it circulates, and that's in a single drop. So you can imagine what's happening inside of me and why I might be leaking out blood and iron. So we're trying to develop that as a test that is more commonly used. Candida is the thing that causes Candida metabolizes sugar and produces uric acid, and this is the effects of Candida on metabolism. It's a mixed bag, but you can see that it's more harm than good. And again, this is just more detail about the effect of Candida. Alcohol is produced by Candida, and alcohol produces acetaldehyde, which is extremely oxidizing. Unless you have alkaline dehydrogenase, you lose it. See, so take your probiotics, eat your vegetables, 80-20 by volume. Minimize release of exposure to heavy metals, toxics, and waste. maintain a nootropic sensitive of biota, minimize compromising agents such as most grains, gluten, and simple carbs, grow and maintain foods high in macro and phytonutrients. This is the new food pyramid. Carbs are gone. They're at the very top. See, it moved from the bottom to the top now. This is the Institute of Medicine Diet Research Panel. This came out just last year. Now you got to exercise, checking your weight, and drink lots of water. It's at the bottom of the food pyramid now. I'll wait for you to take a picture of that. I'm not sure that it made the final cut. I'm afraid Congress might have changed that. So there's a lot of books written on this subject. Eat fat, get thin, eat more healthy fats. Mark Hyman. Eat on the wild side, grow your own vegetables. So when they did this on the International Space Station, they ate their first lettuce sample grown on the space station. It was not lettuce, it was something else, a wheatgrass, I think. They said, that's one small bite for a man, one giant leap for humankind. So on that, I'll end the lecture and see if there's any time for questions.